calorie a calorie? When we think about energy balance or weight maintenance, we recognize that energy intake in the form of calories must equal the amount of energy being expended. In terms of weight loss, one would expect that two weight-reducing isocaloric diets, regardless of macronutrient composition and food properties, would result in an equivalent amount of weight loss, but this is not always the case. Today we'll explore some of the many reasons why a calorie is not always a calorie, both in terms of intake and expenditure. A food's energy can be measured using a bomb calorimeter. When a food is combusted in a calorimeter, the amount of heat produced is measured as food energy. A gram of protein, fat, or carbohydrate contains about 5.7, 9.4, or 4.1 calories respectively. However, the human body is less efficient in extracting these calories than the bomb calorimeter. Adjusting for this reduced efficiency, energy from carbohydrate and protein is 4 calories per gram, while fat provides 9 calories per gram. These numbers are also known as the Atwater factors, and they are frequently used to estimate the energy content of foods. The reduction in the efficiency of energy extraction from foods by the body is influenced by several factors. In this short video, we will focus on a few of these factors, including cooking, chewing, the gut microbiome, as well as the energy costs of nutrient metabolism, which is also known as diet-induced thermogenesis. Let's start with cooking. Cooking has an impact on a food's digestibility and, as a result, the amount of calories it yields. To understand cooking's effect on starch, the structure of starch warrants a brief mention. Starch's structure is comprised of 70 to 80% amylose and 20 to 30% amylopectin. These molecules arrange themselves in a semi-crystalline structure in plants. In starch, cooking disrupts the crystalline structure, and water molecules link themselves to exposed hydroxyl groups of amylose and amylopectin. This results in granule swelling and gelatinization, making the starch molecules more easily available for enzymatic attack by salivary and pancreatic amylase. A study that sought to determine the effects of cooked versus raw starches on their digestibility in growing pigs showed that there was greater digestibility of the cooked potato starch feed compared to the raw potato starch feed. In addition, pigs fed the cooked potato starch feed had significantly larger weight gain per day than pigs that consumed the raw potato starch feed. Does this mean that cooking always alters energy availability from food? The answer is not so straightforward. Other variables govern the release of energy by cooking. One example is the botanical source of the starch. Green banana flour, high amylose maize, and legumes are naturally occurring starches that are less susceptible to digestion in the small intestine compared to other starches. Physical and chemical properties of different plant cell walls also affect the rupture and separation by thermal cooking processes on starch. Consumption of foods that contain amylose and fatty acid complexes, or foods that contain a combination of starch and protein, can also change the degree of enzymatic hydrolysis of starch and energy release. Next, we'll focus on chewing. In a study where humans were given 55 grams of almonds, chewing the almonds fewer times reduced the absorption of fat from the nuts. Specifically, chewing 25 times reduced fat absorption by 11% compared to chewing 40 times, and chewing only 10 times reduced absorption by 13%. In other words, the calorie content of almonds as shown on a food label might not reflect the actual energy obtained by the body. It depends on how well the almonds are chewed. So why is chewing important in absorbing fat from almonds? It has to do with how fat is encapsulated in the almonds. If we look at the cell structure of almonds, fat is kept inside the thick cell wall. When we chew, we mechanically crack the nut and damage the cell wall, making fat accessible to be digested and absorbed. This is why less chewing leads to the reduced fat absorption shown in the study. Next, we'll look at the gut microbiome's influence on energy harvest from food. Our large intestine is populated with trillions of bacteria, which are referred to as the gut microbiome. These bacteria are necessary for the breakdown of non-digestible carbohydrates, which are fermented to yield short-chain fatty acids. The short-chain fatty acids are absorbed and utilized by the host for energy, or de novo lipogenesis and cholesterol synthesis. The composition of the microbiome is determined by a number of factors, including genetics, diet composition, gender, and age. While the data are far from conclusive, a growing body of evidence suggests that obesity is associated with an altered microbiome that is more efficient at harvesting energy from the diet and may contribute to a positive energy balance. Specifically, data suggests that the microbiome alters satiety signals in the brain, short-chain fatty acid production, inflammation, triglyceride incorporation into adipose tissue, fatty acid oxidation in muscle, and the integrity and endocrine activity of the gut epithelium. The best data describing the effects of the microbiome on energy harvest come from animal models. One animal study found that relative to lean littermates, obese OB mice had a microbiome that demonstrated increased enzymatic capacity for energy harvest and decreased energy content in the feces. In a second experiment, germ-free mice were colonized with microbes from OB mice or lean controls. The mice colonized with OB microbes had increased body fat after 14 days, despite no difference in energy intake between the groups, suggesting that this is another case where a calorie is not necessarily a calorie. Recently, a 12-week randomized placebo-controlled trial was conducted to test if probiotic supplementation would affect weight status. This study demonstrated that, compared to the placebo group, the group receiving a lactobacillus supplement decreased body weight and abdominal adiposity. Many more studies need to be conducted, but a growing body of evidence suggests that the gut microbiome may be an important factor in energy metabolism and the prevention and treatment of obesity. Let's explore the other side of the energy balance story with diet-induced thermogenesis. Energy balance occurs when the energy consumed equals the energy expended. Energy expenditure is comprised of three parts: basal metabolism, physical activity, and diet-induced thermogenesis, or the energy expended after eating due to digestion, absorption, and predominantly the storage of nutrients. Today we'll focus on diet-induced thermogenesis to see if a calorie is truly a calorie. Diet-induced thermogenesis accounts for about 10% of total energy expenditure. While this is a small part of total energy expenditure, studies suggest that diet-induced thermogenesis can be influenced by the macronutrient composition of the diet. When measured separately, diet-induced thermogenesis from protein intake elevates energy expenditure by 20 to 30%, alcohol by 10 to 30%, carbohydrate by 5 to 10%, and fat by 0 to 3%. So if we were to consume an equal amount of pure protein, pure alcohol, pure carbohydrate, or pure fat, far fewer calories are left for storage from protein than from fat. Thus, it appears that a calorie from protein does not have the same potential for weight gain as a calorie from fat. While it is interesting to note the differences between individual macronutrients, the reality is that we rarely eat macronutrients in isolation. While combining protein and carbohydrate has been shown to increase diet-induced thermogenesis when compared to just protein alone, the ratio of macronutrients is important. One study showed that diet-induced thermogenesis increased by 28% when the diet was 30% protein, 60% carbohydrate, and 10% fat, versus 10% protein, 30% carbohydrate, and 60% fat. A 28% difference on a 2,000 calorie diet is approximately 80 calories. Maintained over a long time, this energy savings could help to reduce weight. Do high-protein diets result in greater weight loss? Results are inconsistent, with some studies concluding yes and others failing to find benefits. As with many weight loss studies, compliance to the prescribed diet is difficult over the long term. If a calorie is a calorie, our body should receive the exact amount of energy stated on the food label. However, as we've discussed, what and how we eat significantly alters food energy available to our body. It should be noted that there are practical issues related to these findings that likely weaken their real-world application. For example, following a raw diet for weight management is not feasible for most people, and not all foods we eat require chewing. Radically altering the protein content of the diet has been shown to result in poor compliance over time and could result in nutritional deficiencies. The evidence presented to you, therefore, should be carefully considered. There are many research opportunities available through the Ingestive Behavior Research Center, or IBRIC. These opportunities are in areas such as food properties and ingestive behavior, energy and nutrient sensing, endocrine and neural controls of ingestive behavior, energy intake, expenditure, and balance, culture, environment, and ingestive behavior, and ingestive behaviors and health across the lifespan. We hope you'll join us. Studies cited in this presentation include the following: